Welcome to the course Introduction to Complex Biological Systems. In this week 8, I am discussing the evolutionary history of life. So, what we have seen is how evolution shapes the different life forms on this planet. So, today I am going to talk about certain model organisms. To do biological science, what scientists have to use is use certain organisms, right? And it turns out that some organisms are very amenable to the scientific experiments that are done in the laboratory setting. And these type of organisms are typically referred to as model organisms. So today I am going to discuss several model organisms. So introduce their advantages and disadvantages. But one thing that I should point out is this is not an exhaustive list. There are other organisms that scientists also use, but these are one of the most common ones that are used in a lab. So, for doing cellular experiments with eukaryotic cells, HEK293 HeLa cells are used and also Snyder2. So, I will discuss about these cells in more details in the coming slides. For prokaryotic system, E. coli has been a model organism where lots and lots of experiments have been done. For unicellular eukaryotes, budding yeast, that is Saccharomyces cerevisiae, has been used. For animals as model systems, fruit fly, which is Drosophila melanogaster, then a nematode, which is C. elegans, and mouse, these have been the workhorse of biological experiments. So, these are model animals. For plants, these two plants, Arabidopsis and tobacco, they have been used as model plants for scientific experiments. So, this is the tree of life, right? So, these are the different trees of life. So, you will also have viruses, maybe that's as the fourth kingdom, but these are the major three kingdoms of life. And to study each of these kingdom, different model organisms have been used, as we saw in the last previous slide. So, what are the characteristics? Modern organisms are species widely used in research to study biological processes due to their simplicity, ease of maintenance and relevance to other species. So, simplicity means that they can be handled easily. Ease of maintenance means that they can reproduce easily. So, their genetics, all the other characteristics of these organisms are already well known, and whatever information is obtained by experiments in this organism can be translated to other organisms. So, their relevance to other species. Model systems include in vitro techniques like cell and tissue culture that allow controlled experiments in a simplified environment. So, I will discuss this in more details when I talk about the model cells. So, what this type of systems do is that they enable researchers to understand fundamental biological mechanisms such as genetics. So, if you are doing some genetic experiment, you can use this. If you want to study development, how from a single cell a whole life form emerges, you can use these model systems. For disease progression, which is very important, that these model systems are used, and since multiple labs throughout the world use these organisms, there is a consistency in the experiments and reagents have been developed so that they become cost effective. Also, guidelines have been designed so that experiments can be done in an ethical manner. So, using model systems enables all of this. Okay. So, what are the characteristics of these model systems? Why a certain organism has been chosen as a model organism? One of the important characteristics is that they should have rapid life cycle, which means that they should reproduce fast enough so that we can do our experiments relatively quickly. Their genetics should be similar to humans. If we are using animal models, their genetics should be similar to humans, so that if we are doing some experiment in Drosophila or in mouse, we can translate that information into humans. That okay, if this particular protein works like this, most probably it will work in a similar manner for humans also. Availability of complete genome sequence, this is very important. So, we should have the complete genome sequence of this model organism, and we should know how many proteins are there and things like that, right? And there should be genetic tools that are available, because we want to make mutations, we want to delete certain proteins, we want to introduce some other proteins. So, all of these things can be done relatively easy. And normally they can be done relatively easy. That is why these organisms are chosen as model organisms. And also easy maintenance and experimentation. So cells as model organism. So before we get into organism, it is easier to do experiments in isolated cells. There are several reasons for that, because cells can be easily manipulated compared to a whole organism. Cells are easier to manipulate genetically compared to a whole organism. Using cells instead of whole organism reduces the ethical concerns associated with the animal research. So if we are doing some experiment in mice, the ethical considerations for doing experiment in mice versus ethical consideration for doing an experiment on a isolated cell, they are completely different. In cell, the ethical considerations are much simpler. So one can do experiments with rapid experiments without worrying too much about all the ethical concerns, right? Large number of cells can be cultured. So there is a limitation in the number of mouse that you can use in a particular experiment. But when we are doing experiments in a cell, we can grow large number of cells in a cell culture. And this type of setting can be used for high throughput screening, for example, drug screening. So if you are trying to inhibit some protein or an enzyme, like we saw in the previous lectures, and there is a library of 10,000 molecules. So a cell culture or isolated cells will be much better to start with to see whether this particular drug has the desired effect on the enzyme inside the cell, rather than looking at the whole organism. If it works at the cellular level, then it makes sense to go for the organism level. So this is how the hierarchy of experiments are typically designed. Cells grown in vitro allow researchers to focus on specific biological mechanisms without interference from the complexity of an entire organism. For example, if someone is looking at the effect of a particular molecule on liver cells. Now, if we are doing with cultured cells, that molecule can be directly delivered to the cultured cells. However, if it is ingested, so if it is given in food and the mice digest that food, then it goes through its digestive system. So it will go through a lot of other environment before it reaches the liver, right? So if we are doing cell culture, we don't have to worry about all these processes that are there before the molecule reaches the liver. However, you will have to worry about all those things once your cellular experiments are successful. Then you will have to go to the organism level. So let's look at some model cellular systems. HEK 298, so HEK stands for human embryonic kidney. So this human embryonic kidney 298 cells, they were derived in 1973 by Dr. Alexander van der Ebb. These cells are very useful and they are routinely used in different cell culture experiments throughout the world. They can be easily transformed using shared adenovirus, giving cells that are stable and they are immortalized. So they have an immortalized phenotype, which means that this particular cell line can be used again and again in all these different labs. What are the advantages? They are widely used for recombinant protein production. In genetics, so in recombinant DNA technology, you will learn that we typically use bacteria to produce protein recombinantly. So that is the first choice. However, there are many proteins which do not express well in bacterial systems, especially if those proteins have post-translational modifications. So in those situations, cells like HEK 298 can be used for overexpression of these target proteins. So this is also, it also has high transcription efficiency, which means that silencing experiments can be also done very relatively easy. Disadvantage, so it is derived from embryonic kidney cells, so they are not fully representative of the specific tissue or primary cells. So since these are embryonic kidney cells and these are isolated cells, plus they have been immortalized, they do not resemble the primary cells from the kidney. Also, they will not resemble the actual cells inside the organ. And the third point is that they will be different from other cells. So if you want to do experiments in lung cells, then HEC298 cells will not be good enough. So you will actually have to take cells which are derived from lung cells. So one has to be aware about the tissue specificity when they are using systems like this. Another extremely commonly used cell line is the HeLa cell line. So this cell line was derived from the cervical cancer cells from this woman named Henrietta Lacks in 1951. And since it was such a long time back, you can imagine that not much ethical guidelines were there, so it were actually taken from her without her knowledge or consent. However, it turns out that these cell lines are highly robust, these cells are highly robust, and they are an immortalized cell line, so they can be used again and again and again. So HeLa cells have been used in many breakthrough research over the past 70 years. So it has contributed to breakthroughs in cancer research, in virology, and in molecular biology. So it is a model for evaluating potential therapeutic agents and toxicity. So you will see that in many labs, one of the first experiments that people do will be done in HeLa cell line. Disadvantage, they are cancerous cells, right? Which means that they do not represent the normal cell behavior. But many experiments can be done using these cell lines as long as you are aware about these differences. Another very commonly used model cell system or cell line is Snyder 2. So this is derived from embryonic tissues of Drosophila melanogaster, the fruit fly. So it was isolated in 1972 by Dr. Irving Snyder, and it is named after this inventor. These cells are also immortalized, they are non-adherent, and they grow as a semi-suspension in culture, which means that you can actually use them in some shaker kind of environment. So these are also cells which are very robust system for expression of recombinant proteins, especially if those are coming from a Drosophila or some other organisms like this. So these are widely used to study Drosophila gene function and regulation. Since it's an insect cell line, findings may not always directly translate to mammalian or human system. So one has to be aware about the limitations of this particular cell line. So these are the three cell lines that are derived from eukaryotes. Prokaryotes, also there are many studies that we do in prokaryotes, so we also have to have a model system from prokaryotes, right? So prokaryotes such as bacteria, they serve as great model organisms in basic research. So prokaryote cells are structurally and genetically simpler, so it is much easier to do experiments with them. E. coli is a model organism that has been used to study prokaryotes. So they have very short generation time, which means that they can multiply very fast in a very short period of time. They can be easily transformed with foreign DNA, allowing genetic modifications. So tools like plasmid, transposons, and CRISPR systems are routinely used in prokaryotic models. They grow on simple, inexpensive media. So growing a bacteria is much more inexpensive compared to growing a eukaryotic cell like HeLa or Snyder 2. So E. coli is a model prokaryotic organism. It is a gram-negative bacteria. It is facultative anaerobic, which means that in the presence of oxygen, it will use oxygen to produce ATP. So it will go through the aerobic process. However, in the absence of oxygen, it will go to the anaerobic metabolism, anaerobic process. It is rod-shaped, so you can see its rod-shaped like structure here. So E. coli has been a workhorse in biotechnology for the production of recombinant proteins, enzymes, and metabolites. It is widely used for cloning, gene expression, mutagenesis, and metabolic pathway analysis. It is instrumental in the discovery of the genetic code. So we have looked at genetic code. We have looked at which codon codes for which amino acid. So those discoveries were made in E. coli. Another important discovery that was made was the operon model. So the lac repressor model that was also discovered in E. coli. Disadvantage, of course, it lacks organelles and eukaryotic specific processes. So it can serve as a model system for basic research or research that are suitable for prokaryotes, but not for eukaryotic systems. So this is a brief description of the lac operon. So you have already seen that. So this lac operon system was discovered in E. coli by Jacob and Monod in 1961. So, briefly, in this repressor, the lac repressor comes and binds, and once there is lactose in the system, then this repressor, it gets converted to allolactose, it falls off, and these genes get translated. So, this particular system where consecutive genes are regulated by a single promoter is called an operon, and it is found only in prokaryotes. So, this system was discovered in E. coli. Now, there are also eukaryotes which are unicellular. A model system for unicellular eukaryotes is Saccharomyces cerevisiae, that is Baker's yeast. Another model system is Tetrahymena thermophilia. Again, these model systems have been used to make many groundbreaking discoveries. For example, Tetrahymena was used for the discovery of RNA splicing. So, RNA splicing was actually discovered using Tetrahymena. Saccharomyces cerevisiae, I will show one example in the next slide, which is the yeast to hybrid assay, which is done using Saccharomyces cerevisiae. So, these type of eukaryotes, they have processes which are very similar to the multicellular eukaryotes because they have all these internal organelles. So, they can serve as a model system for studying eukaryotic systems. Since these are unicellular, they grow fast and they can be cultured in large quantities in relatively inexpensive manner. So, many key cellular mechanisms such as cell cycle, DNA repair, transcription, translation, etc. are conserved in these unicellular eukaryotes as that of higher eukaryotes. So, Saccharomyces cerevisiae, commonly known as the budding yeast, is a widely used model organism. It has been used to study checkpoint controls, cyclin dependent kinases. So, these things that we have seen in the previous lectures, they were widely studied using this model system. It is also a well established model for studying metabolic processes including fermentation and respiration. Its ability to perform post-translational modifications such as glycosylation also makes it a suitable system for expressing complex eukaryotic proteins. So, proteins which need glycosylation, you cannot express them in bacteria, but you can express them in yeast. So, what are the disadvantages? Since it is a unicellular organism, it lacks organ system and it lacks more complex biological systems such as the immune function. So, you cannot study immune system using this type of unicellular organism. Another important experiment that was first done in yeast is the yeast to hybrid system. So, this is a very useful experiment that is used to study whether two proteins interact with each other. So, this particular protein GAL4 binding domain and this activation domain, they are normally linked together. So, what was done is that they have been separated, and now using recombinant DNA technology, so you want to study the interaction between this protein which is called the bait and this protein which is called the prey. So, you want to screen whether these two proteins interact with each other or not. So, you genetically fuse these two genes together and fuse these two genes together, and then you express them. If they interact, then bait and prey come together. So, they are binding tightly. GAL4, the binding domain, it will bind to this upstream system. So, when it binds to this is linked to it, this binds to this is linked to it. So, it will bring in this activation domain and it will activate the transcription of this reporter gene. This reporter gene can be anything. It can be a marker for an antibiotic resistance, which means that when this interaction happens, those cells where this interaction happens, they will survive because this protein is expressed, and if you have given the antibiotic in the media, it will be able to survive. If there is no interaction, then there will be no survival. Or it can be a fluorescent molecule. So, if there is interaction, there will be fluorescence. If there is no interaction, there will be no fluorescence. So, using this type of systems, we can screen for molecules that interact with this bait. So, if you expect 20 different proteins that may or may not interact with this protein, you can clone them separately. So, you can have them separately and then transform them and then do this experiment. and then see which proteins are interacting. If you want to identify which residue, suppose I have these two proteins, I know they interact, but I don't know what is the binding interface. So you can mutate residues at this binding interface and screen it using this. So you can screen all the different mutations using this type of system. So anything that survives, you know that is needed. So if you mutate some residue and if it does not survive, you know that that particular mutation is needed for this interaction. So there are many ways these experiments can be done, right? So yeast actually contributed to the development of this type of experiments. Now, animals as model organisms. So we have looked at single unicellular organisms, we have looked at prokaryotes, we have looked at eukaryotes. Now, animals as a model organism. So once you have done your experiments in the cellular environment and you find interesting results, now you may move on to animal models. So animal models provide a system for studying complex interactions between different tissues, organs and systems, which cannot be easily replicated in cell cultures or simpler organisms like yeast. Many animals, such as mice, can be genetically modified through techniques such as gene knockout, transgenics, CRISPR-Cas9, etc. So this type of techniques can be used to model certain systems. For example, if you want to study Alzheimer's disease, you can introduce certain mutations which will make the mice predisposed towards the Alzheimer's disease. So now you have a mouse model which is an Alzheimer's mouse model. And then you can screen for different molecules or different therapy to see whether they are able to reduce the effect of dementia in this particular mice model. So all of this type of genetic manipulations can be done in this type of model organisms. Animals are used to study complex behaviors such as learning, memory, social interaction and emotions, which are challenging to study directly in humans. So if you want to study learning, what are the proteins which are important in short-term memory or long-term memory, then this type of animal models can also be used. Animal studies play a critical role in preclinical testing for drug development. So of course, after the cell, you go to these different animal models to see whether these drugs are actually effective in an organism setting. If they work, then of course you go to higher organisms and finally to human clinical trials. So let's look at fruit fly or Drosophila melanogaster. So Drosophila melanogaster or fruit fly is one of the most widely used model organisms in biological research. Its contribution spans to genetics, developmental biology, neurobiology and many more. So a lot of research, early research on developmental biology were done in fruit fly because of its ease of genetic manipulation and very fast life cycle. So Drosophila, they have a short life cycle of 10 to 14 days at 25 degrees centigrade. They have a very high reproductive rate and clutch size, which means that there is enough material to do experiments. Drosophila genome is small. It has only 4 pairs of chromosome which are fully sequenced, and there are extensive genetic tools which are available that can be used to manipulate the Drosophila genome. Many biological pathways and genes in Drosophila are conserved in humans, making it an excellent model for studying human disease. For example, if you want to study development, then the same set of genes are present in Drosophila as well as in humans. So if we think of the Hox genes, there are 8 Hox genes in Drosophila, there are 39 Hox genes in humans, right? Even though the number of genes are different, but the basic principles remain the same. Disadvantages: difference between Drosophila and humans in physiology and organ systems, for example, absence of adaptive immune system. So these things make it difficult to study certain phenomena in Drosophila which are important for humans. So in that case, you go to a different model which will be a mice model. So Drosophila has contributed to many discoveries. One of the important discoveries that happened in Drosophila is the chromosomal theory of inheritance. So Thomas Hunt Morgan, he was an American zoologist and genetist. His famous experiment on Drosophila established this chromosomal theory of heredity. So he showed that genes are linked in a series on chromosomes and are responsible for identifiable hereditary traits. Okay. So another very important discovery that happened in Drosophila is the homeotic transformation. So it turns out that there are these master regulated genes called the Hox genes. So in Drosophila there are these 8 Hox genes: labial, deformed, sex chroms reduced, antennapedia, ultrabithorax, abdominal A and abdominal B, and they are arranged in this linear order on a chromosome. And it turns out that each of these genes regulate the development of each of these segments in the body of this Drosophila. Now these genes are also present in us and they do exactly the same in us also. So this gene is responsible for the development of the head, so on and so forth, right? Now there is also another very interesting discovery that was made, which is that if you copy one of these segments and add another copy, then the same thing will be duplicated. So for example, what happens here is that the Drosophila has 2 wings and there are these small structures which are called halteres. So they help in balancing when they are flying. Now a particular set of genes was copied which resulted in the formation of 2 pairs of wings. So instead of these halteres, there were another pairs of genes that pairs of wings that was found in this mutant Drosophila. In fact, the position of these genes are also important, which is called the collinearity of these Hox genes. So in this case, antenna, so from the head of the Drosophila, this antenna they come out and they are normally determined by this antennapedia. Okay. Ultrabithorax is responsible for the development of the leg. Now in one experiment, this gene was deleted from here and it was inserted before the antennapedia. So in those mutant flies, what happened is instead of this antenna, legs came out from the head. So it means that these genes are highly modular in nature and by shifting the position, the body plan can be changed. So this was a very important discovery in the development of vertebrates. Another important organism is a nematode, which is C. elegans. So C. elegans is a microscopic nematode or roundworm. So it is very small, much smaller than mice and Drosophila, and they are widely used in biological research. So it is 1 millimeter long. It contains roughly 1000 somatic cells. Yet it has a fully developed nervous system, digestive system and a reproductive system. Now since it is such a small organism, the lineage of all these cells are completely known. So this organism is also transparent. So if you put it under light microscope, you can see all the cells, you can actually see through this organism. So its transparency allows researchers to easily observe cellular and developmental process under a microscope. C. elegans was the first multicellular organism for which the entire cellular lineage was mapped. So from a single fertilized egg, one cell becomes 2 cells, then it becomes 4 and so forth. Where each cell goes, which cell is responsible for forming the anterior side and which cell is responsible for the posterior side, all of this was mapped for the first time for C. elegans. Discovery of key apoptosis genes, for example CED3, 4 and 9, they were done in C. elegans and it provided important insights into the genetic control of cell death, that is apoptosis. So what are disadvantages? It lacks a complete complex systems which are found in higher organisms such as adaptive immunity. So again, if you want to study something like that, then C. elegans is not the model organism. You have to go for something like mouse. So another important discovery that was done using C. elegans was the phenomenon called RNA interference. Andrew Fire and Craig Mello, they demonstrated that injecting double-stranded RNA, not single-stranded, but double-stranded RNA was very effective in silencing specific genes. And this was done for the first time in C. elegans. It was also shown that this effect was heritable, which means that subsequent generation of the worm also exhibited silencing of the target gene. So, a very important discovery. This is another important discovery that was made using C. elegans. Now, the most widely used organism is the mouse model. So, the house mouse or mus miculus is one of the most widely used model organism in biomedical research. So, it has many advantages. Almost 99% of the mouse genes are homologous to human. So, again if I talk about the Hox genes, there are 8 Hox genes in Drosophila, but 39 Hox genes in humans, and if we see mice, they also have 39 Hox genes, and these genes are very identical to the human genes. So, it means that to study development in humans, the developmental study in mice can give us much more detailed clue. Okay. So, mouse models mimic human diseases such as cancer, diabetes, cardiovascular diseases, neurodegenerative disorders such as Alzheimer's and Parkinson's disease, and also infectious diseases. Mice are typically used for preclinical testing of drugs and to study toxicological effects of various compounds. So, once you have screened a set of molecules using cellular system, and if you have come out with, let us say, 4 or 5 target molecules, then those will be tested in mice to see the effect on an organism level. And once that is successful, then typically we will go for higher animals such as rabbits or guinea pigs, and then dogs, pigs or primates. Mice, they are also used to study complex behavior like learning, memory, anxiety, and also social behaviors. So, neuroscience, to study neuroscience, mice is also a very good model system. Disadvantages, well, it has of course these ethical issues. However, there are many things that are studied in mice cannot be directly translated to humans. So, that also poses a big disadvantage in using mice as a model system for studying really complex systems. So, one very important discovery, there are many discoveries that have been done using mice, but one important discovery that needs mention is the discovery of induced pluripotent stem cells. So, I have already discussed this earlier. So, iPS cells or induced pluripotent stem cells were discovered by Yamanaka and Takahashi, and they did that using adult mouse embryo. So, they showed that these 4 transcription factors, OCT4, SOX2, CMYC and KLF4, when you add them, you can actually turn somatic cells into these induced pluripotent stem cells. Now, for mice, so mice is one of the model organisms in animals, but there are also model organisms for plants, right? So, in case of plants, many plants are easy to grow, reproduce rapidly and require minimal resources. So, many plants can be used for used as model organisms. Unlike animals, plants raise fewer ethical concerns in experimental research. Plant models are necessary for research concerning improvement in agricultural sector, which is a very important concern for us, so that we can generate high yielding crops that can tolerate different stress conditions and are pest resistant. Plant models have relatively simpler genome compared to animal models and are amenable to genetic modifications. So, one of the model plants that are used in many labs is the Arabidopsis. So, it is one of the most widely used model organism in plant biology for the following reason. It is small, it has a fully sequenced genome, high efficiency for genetic transformation using agrobacterium. So, large collection of mutants and transgenic lines can be easily produced for this particular plant. Its complete life cycle is 6 weeks, which makes it a rapidly growing plant which is very useful for doing experimentation. It is a dicotyledon, so it is a dicot, which means that it shares many features with economically important crops. However, findings in this dicot plant may not directly translate to monocot species like rice or wheat, which means that for studying rice or wheat, we have to use model organisms or model plants from this particular species. Another very commonly used plant is the tobacco. So it has been a prominent model organism in plant biology due to its versatility, ease of manipulation, and historical significance. So it also has very high transformation efficiency, and tobacco, due to its large leaves, so you can see that the leaves are very large. It is a leading system for chloroplast transformation, enabling high levels of protein expression in these organelles. Tobacco mosaic virus, or TMV, has been a vital tool for studying viral infections and plant immune response using tobacco as the model organism. So immunity in plants have been studied using this tobacco plant system, where tobacco mosaic virus have been used to induce these viral infections. And there are, of course, disadvantages. For example, it's an allotetraploid, which means that it has a complicated genome, which makes its genetic analysis complicated. So tobacco mosaic virus, it turns out that it was the first virus to be discovered. So the infection causes characteristic patterns such as mosaic like mottling and discoloration on the leaves of tobacco. This is a picture. So this is a big tobacco leaf, and infection makes it look like this. That is why it was called tobacco mosaic virus. So in 1982, Dmitry Ivanovsky, he gave the first concrete evidence for the existence of a non-bacterial infectious agent, showing that infected sap, which was extracted from these leaves, infected tobacco leaves, but even though you filter this sap through a filtering agent, it can still infect these tobacco leaves, which means that there is something which is even smaller than known bacteria that can infect, and that is how the first virus was discovered. So here are the conclusion. Model systems where cellular models or whole organisms are used, so they simplify the study of complex biological systems. They provide insights which are applicable to other organisms. So they apply broadly due to their conservative genetics, molecular and physiological processes across species. So whatever we learn from model systems, we can translate that information to other systems, and they can also contribute to basic or fundamental research. Now these models, organisms or systems, they offer ethical and practical alternatives to studying higher organisms. So there are many ethical issues which come with actual organisms like mice or Drosophila. However, if we are using isolated cellular systems, then we can circumvent some of these ethical issues. Different models are suited for specific research, as we have seen in the previous slides. So here are some of the important references, and you can also refer to these books for this particular lecture. Thank you.